So, resuming from where I left in the previous lecture, unfortunately, I was cut out by the tool that I used to record the lectures. So, we talked about Mendelian genetics. And so, obviously, these genetics extend to many characters found not only in plants, but also in humans. Here you have some examples. They're given by your book. Some of them actually have mistakes, like the web fingers are actually dominant, not recessive, and so on. But these, what I want to tell you is that the Mendelian genetic extends also to many other characters in different type of organisms as well. There are also exceptions though. And one example is given here. So there are many exceptions to Mendelian genetics. One is when you have incomplete dominance of alleles versus the other. What Mendel had stipulated is that one allele is always dominant versus the other, right? You had the purple being dominant over the white when it comes to the color of the flowers in pea plants. However, this is not always true. Sometimes you have that some alleles are partly dominant, not completely. Here is an example. If you look at the flowers in this plant, which is called snapdragon, you have that now. If you do the same experiment that was done by Mendel, where you start with true breeding, red plants, and white plants. So you have two possible here, red and white. Now, when you cross them together and you produce a heterozygous plants, at this point, you don't have these plants with having just one color identical to the one of the parental plants. What you have is that, in fact, in the heterozygous plants, the color is almost like more of a blending. So red is still dominant over white, but not completely dominant. It gives you a lighter color when it's heterozygous. And you can see this even when you take these plants and as Mendel did, you let them self-pollinate or cross-pollinate, producing the F2 generation. There too, you'll see that you get red plants identical to the original parental plants, only when you have homozygous dominant plants. But if you have heterozygous plants, they'll continue being like some sort of mix between the red and the white. So in this case, red still imposes itself over white, but is not completely dominant, giving a color, lighter color when it's found in the heterozygous form. So this is an exception to Mendelian genetics. Other exceptions are sometimes you have that alleles, they're not incompletely dominant, but they're non-dominant at all. You have that all alleles dominate the same way. One example is when you do the M and N blood typing of people. This corresponds to proteins, glycoproteins, I should say, found in the red blood cells, which are presented in red blood cells. So if you have the, let me just draw it here. They are found as on the surface of red blood cells here. Let me just do the pen. So if you have, these are your red blood cells, you could have both the M. So if you have the M allele, you will produce this M glycoprotein. If you have the N, you will produce. Both of them will be present. None of them is dominating over the other one. So you could have, if you are heterozygous, you would have both, like in this case. Or if you are homozygous for M, you would have only M present there, right? These will all be M. The same thing for N, but none of them is dominant. If you're heterozygous, you have both of them present there. There is no dominance. You refer to this as codominance, differently from what Mendel had stipulated. Same thing, sometimes you may have multiple alleles, not only two. Mendel's always work with two alleles, two possible variables there, but you may have more of them. And you can see when you do genetics based on changes in single nucleotides in the DNA, you may have different variants there. One example is when you look at the A and B blood typing. Now you have three possible alleles there. You can have an allele that gives you the A, so a gene, a copy of the gene that gives you the A protein, one that gives you the B proteins, and one of them that doesn't code for anything. That's why it's referred to as zero. So you can have all the possible combination there. You can be homozygous for A, you can be heterozygous for A and B, or you can be heterozygous for A and the gene that doesn't code for any antigen. So you may have multiple possible combinations there other than those based on two factors there. So you may have more than one alleles present, unlike what Mendel had studied. Another big variable is the one based on epistasis. We talk about epistasis when you have the one character is actually dictated by one gene, which, however, is under control of another gene. One example is the colors of the fur in these beautiful dogs called Labradors. Now, if you have noticed, you can have actually three different types of colors in Labradors. You can have black labs, beautiful. You can have chocolate labs, intermediate, beautiful too. And you can have the light color one, which are beautiful. All of them are super cute. However, how is their color determined is by how much melanin, this pigment, is produced on their fur. Okay, and the production of the melanin is done by a gene. Here it has two variables, capital B, dominant over small b, where capital B would determine the production of a lot of melanin, and B would determine, recessive B, would determine less melanin. But both of them would have melanin. This will be actually the picture that you get if these two alleles were the only one controlling the situation. But now you have a second gene in the picture, which is the one shown as capital E or small e, which actually control whether the melanin will be deposited or not. So if it gets deposited, that's the two possible scenarios you can get. If it doesn't get deposited, which is the recessive form, you got actually the yellow form right here. So look at this cross here. For example, if you take two true breeding, let me erase a little bit of stuff here. Sorry, I like to go crazy with this. Let's see if I can get erase it. No, that's too much. Okay, so but look at the cross that you have right there on the bottom. So if you take now two dogs here. So you take a black dog, which is homozygous dominant for both genes, and a yellow dog, which is homozygous recessive for both. Now, if you looked only the B gene, actually this dog will be black, this should be brown, because a small b still allows for the production of melanin that will make it brown. What makes the difference is the second gene here. This gene is homozygous dominant for the black dogs, which basically says, okay, the melanin could be deposited in the fur. It's different in the nose. And this is homozygous recessive, which says no melanin should be produced, all right? So, which means even though you should have a brown color like this, the intermediate one, there is no melanin allowed to be deposited in the fur, and therefore the dog will look much lighter, light yellow. When you mix them together, you will get a generation here, the F1 generation, which is heterozygous for both of them. So it would be dominant, or heterozygous, for the production of melanin, which basically would dictate a lot of melanin be produced. And it's heterozygous so for the second gene, which still says, having a capital E, still say, dominant form says, okay, the melanin can be deposited. So you produce a lot of melanin, can be deposited, you get a black dog. All the dogs are black. Now you take these heterozygous dogs and you cross them together. All right, so the possible combination of these two genes in the gametes are the one right here. So you take two of these heterozygous dogs and you cross them together. And now you see all the possible combination. Basically, what you get is that if you have a dominant form of the E gene, you would have the pigment produced, right? So whether you are in this situation, B or B, capital B, or capital B and small b here, you would have that the pigments are produced, right? Lots of pigments are produced as long as you have one capital E is allowed to deposit, all right? And so you will get black dogs, okay? Now the scenario is different if you have this situation. If you are in a situation where you are homozygous recessive for the amount of melanin that you produce, if that was the only gene, you will be brown, right? Because you're producing less melanin, okay? So you wouldn't be black, you will be brown. However, you have two scenarios. If you are in this scenario right here, not you, but the dog, now the small amount of melanin can be actually released on the fur, and therefore you get brown dogs, less melanin, but it's still there, produced, right? But now, if instead you have the second gene being both in the recessive form, this blocks the release of melanin, and therefore you will get this beautiful, beautiful yellow dots. Okay, so this again is another example of non-Mendelian genetic, referred to as epistasis, where you have that one gene controls the character, but there is another gene that controls whether the original gene can act or not, like in this case. Other variation from Mendelian genetics basically says that one gene controls one character. This is not always true. There are characters that are controlled by many genes. Okay, and these are referred to as polygenic type of characters or polygenic inheritance. An example is the height, the skin color, which I always use, the body weight, and so on. They are determined by different type of characters, different genes, sorry. And these, you can see it, for example, in the distribution. So these would not give two clear phenotypes, one color or the other one, and so on, one weight or the other weight, or one height and the other one. All the different phenotypes will be distributed following a bell curve, like in this case here for height and so on. So some characters are determined by more than one gene, so they are polygenic type of characters. And then you have also a single gene can also control different characters, like in this example. In this case, you will refer to as pleiotropy. That's where you have a single gene that again dictates different characters, separate characters, like for example sickle cell disease, which controls the shape of the red blood cells, at the same time controls whether you are prone to have mental impairment, heart failure, and so on. These are all different characters, again controlled by the same gene that causes sickle cell disease. All right, so we're gonna stop here, and then this is the last lecture. Hope you enjoyed it. All right.